Translation is the process by which the genetic information encoded in messenger RNA, mRNA, is decoded to produce a specific sequence of amino acids, forming a protein. Translation occurs in the cytoplasm of the cell, specifically on ribosomes, which serve as the sites of protein synthesis. Output: translation begins with the binding of mRNA to the small ribosomal subunit. The initiator tRNA, carrying the amino acid methionine, MET, binds to the start codon, AUG, on the mRNA. The large ribosomal subunit then joins, forming the initiation complex. During elongation, the ribosome moves along the mRNA strand in the 5-to-3 direction, reading each codon, 3 nucleotides, and recruiting the corresponding tRNA molecule carrying the complementary anticodon and amino acid. Peptide bonds form between adjacent amino acids, catalyzed by peptidyl transferase activity of the ribosome. The ribosome translocates to the next codon, releasing the empty tRNA from the P site and shifting the growing polypeptide chain to the A site. Scene script: translation terminates when a stop codon, UAA, UAG, or UGA, is reached on the mRNA. Release factors bind to the stop codon, causing the release of the completed polypeptide chain from the ribosome. The ribosome dissociates into its subunits and the mRNA is released. After translation, the newly synthesized polypeptide may undergo various modifications, including folding into its functional 3-dimensional structure, cleavage of signal sequences, addition of chemical groups, for example phosphorylation, glycosylation, and assembly with other polypeptides to form multi-subunit proteins. The genetic code is degenerate, meaning that multiple codons can code for the same amino acid. However, each codon specifies only one amino acid, and the genetic code is nearly universal across all living organisms. Translation can be regulated at various stages to control protein synthesis in response to cellular needs and environmental cues. Regulatory mechanisms include modulation of mRNA stability, translational efficiency, and post-translational modifications of translation factors. Understanding DNA translation is essential for comprehending how genetic information is converted into functional proteins, playing a critical role in cellular processes, organism development, and disease mechanisms.